The topic for today's session is sterilization and disinfection. Now, these two terms are similar in terms of their action, but they are distinct in how we are bringing about the techniques to eliminate a particular microorganism. Now, as we all know, these microorganisms are ubiquitous in nature. That is, they are present everywhere. You can see them in air, you can see them in the soil, everywhere. So, these organisms, though they are present everywhere, they are sources of different types of infections, they are sources of different types of contamination, they are source of different types of spoilages that they bring about in the food material. So the main objective of sterilization and disinfection is to eliminate these microorganisms. So in microbiology, these procedures, that is sterilization and disinfections, are mostly used for three purposes. One is to prevent contamination, next is to prevent decay of the food or spoilage of the food, and third is to prevent sepsis in particular surgery, that is to maintain aseptic conditions. So this is the most important purpose of sterilization or disinfection in microbiology. So the method that we are adopting for sterilization depends on the material on which we are going to sterilize, the type of organism we are going to eliminate, and depends upon the nature of organism that we are trying to eliminate. So here, coming to the definition of sterilization, sterilization is complete elimination of microorganism from a particular substance. So the objective is to eliminate an organism from a particular substance because they are sources of contamination, they are sources of decay, they are sources of infection. So coming to the term disinfection, it is removal of microorganisms that are pathogenic in nature, that causes different types of infection in human body. So mostly these infections are applied on the human body, are used up in the human body to eliminate the pathogenic microorganism, whereas sterilization is removal of organism from a particular object. So any object that is made completely sterile, that is removed of microorganism, will see to it that there are no microorganism present on that particular object. And one thing that should be remembered over here is a particular object, when it is made sterile, it is completely sterile or it is non-sterile. We cannot say that it is a semi-sterile one because organisms are completely eliminated or they are not eliminated. It can never be semi-sterile. So the two terms, sterilization and disinfections, though they have the same objective, but on the material on which we are applying is completely different. So sterilization in case of a particular object is to completely eliminate or kill the microorganism and make a substance free of microorganism, whereas disinfection is to eliminate the pathogen from the human body. And there is another term that is used in disinfection or relation to the disinfection, that is called as antisepsis. That is nothing but inhibit growth of microorganism from the skin. So these organisms which multiply on a particular skin has a property of forming pus or causes septic. So in order to eliminate this, we use variety of disinfectants. So that is called as antiseptics. And the mode of action of this sterilizing agent or disinfections are mostly, they are either bactericidal or bacteristatic. Bactericidal are the one or the substances that completely kill the microorganism. Bactericidal, cidal is to kill the organism. They completely eliminate the organism. Whereas bacteriostatic are the compounds that inhibits the growth of microorganism. Inhibiting the growth of microorganism means they are not allowing the growth of microorganism on a particular substrate. So you cannot say that these bacteriostatic compounds are sterilizing agent because we are not completely killing the microorganism or removing the microorganism, but we are trying to inhibit the growth of the microorganism. So the mode of action of certain agent is that either they are bactericidal or they can be bacteriostatic in nature. And another term that is used in this sterilization or disinfection is called as decontamination. Decontamination is complete removal of microorganism and other chemicals or radioactive elements from a particular object. So in this particular introduction, we have seen what is sterilization, what is disinfection. Though they are similar, but they have distinct properties, and the objective is to completely eliminate the microorganism. Now coming to the different mode or different ways in which we can bring about sterilization. So as I told you, in microbiology, it is the three purpose for which you usually go for sterilization. One is to prevent contamination, to prevent spoilage of the food, and to remain or maintain aseptic condition. So here the type of sterilization that we are adopting depends upon three important things. One is the type of material that we are sterilizing, the nature of the organism, and on which we are going to apply. So the agent of sterilization are selected depending upon these three purposes. So they can be basically classified as two types, that is a physical modes of sterilization and chemical mode of sterilization. Under physical mode of sterilization, the things that are usually taken up are sunlight, drying, dry heat sterilization, and moist heat sterilization. So you will know in the later part how these heat can be applied in the form of a dry heat or a moist heat. So in the dry heat sterilization, we usually go with the incineration or hot air oven, and in the moist heat we go for pasteurization, boiling, and steam autoclave or autoclaving. And the other method of physical sterilization is radiations and ultrasonic and sonic radiation. So these are all nothing but the physical agents that are used in physical methods of sterilization. Now coming to the chemical method of sterilization, we use wide variety of chemicals in order to bring about the sterilization, and mostly these chemical agents are nothing but the disinfectants. They can be applied either on the human body to reduce the infection, or they can be used on any cleaning surfaces where we want to eliminate any type of microorganism. So under chemical mode of sterilization, we have got wide variety of chemicals that have bactericidal as well as bacteriostatic nature. So the most prominent one being the alcohols that are mostly used in the laboratory to eliminate the organisms, and we have aldehydes, we have dyes, different types of dyes that have bactericidal activity, we have halogens, and we have phenols, surface active agents, metals, and gases. So these are the different modes of sterilization that can be used to eliminate organism from any surfaces. So coming to the physical methods of sterilization, the first one being the sunlight. So sunlight is the most cheap and less expensive form of sterilization. So sunlight has different radiations that has the ability to kill the microorganism. The most prominent one being the ultraviolet rays. So this ultraviolet rays has deleterious effect on microorganism. When the organisms are exposed to this ultraviolet rays, they usually get killed due to the ultraviolet rays acting upon the nucleus, that is the DNA molecule. So these ultraviolet rays are called as non-ionizing radiation. They go and bind to the DNA, form thymine dimers, which are the most important component of a DNA. The thymine dimers inhibits the replication of the organism, as a result of which the organism is killed. So sunlight is the most important and the most cheap, you can say, or less expensive method of sterilization. And the type of rays that are falling on the organism, the type of condition, the type of environment also shows the efficiency of this sunlight rays killing the microorganism. Take for example, if the area has less pollution, the sunlight can directly fall on the organism and kill the organism. Whereas if it is polluted, then these pollutants usually inhibit the rays to fall on the microorganism, and they will inhibit the killing of the microorganism. So the first method is sunlight, that is a natural source that can kill the organism in a natural environment. The next method of sterilization is called as drying. Drying means elimination of water from the organism. As already discussed in the previous topic, moisture is the most important component of a particular organism. So organism is made up of 80% water plus 20% of other component. The water is very, very important to carry out various biological activities inside the organism. So once this water is removed, the organism gets dehydrated and it has no longer any water molecule to carry out its biological activity. So all the vegetative cells can be easily killed with the help of dehydration, that is removal of water from the organism. But this dehydration part will not kill the spores, because spores are highly resistant form. They are not killed by the dehydration. Now the spores are the one that are formed during unfavorable conditions. So they can survive for a period of 400 to 500 even without the presence of water. So once the return of natural conditions or favorable environment is there, the sporulating forms can generate into the vegetative form. So drying does not have any effect on the microorganism that are sporulating in nature, but we can easily kill the vegetative forms of microorganism. Now the third method that we usually adopt in bringing about sterilization in the lab is the heat. Heat is an important method of choice, or the most reliable and the best choice to bring about sterilization or to completely eliminate the organism from a particular environment or particular surface. So depending upon the material that we are going to sterilize, whether it is a heat sensitive material or a resistant material, we have to apply the temperature. So there is a time temperature relationship that we have to know. So if the material is sensitive, then you have to take up the temperature as a short less temperature for a longer period of time. And moreover, if the material is resistant to high temperature, then you can increase the temperature but for a short period of time. So time and temperature is inversely proportional to each other, where if you are applying high temperature, go for a short period of time. If you are applying low temperature, then go for a longer period of time, or you can go for repeated cycles to eliminate the microorganism from a particular object. Now coming to the mode of action of the heat or any method that we are using, mode of action is how they usually target a different component in the organism. So there are variety of components that are present in the organism that helps it to grow and reproduce. So these sterilizing agents usually go and target certain components in the organism, thereby rendering it inactive or completely killing the microorganism. So the mode of action of different agents brings about the killing of organism. So here in the case of heat, the mode of action is the protein denaturation. Protein is an important component of any organism that helps us to carry out different biological activities. So the protein is the one that has been inactivated by the heat. So it causes the denaturation of protein. The quaternary structure of the protein that is functional in nature is completely denatured by the heat. As a result of which, the organism cannot carry out its biological activities and thereby it gets killed. Apart from that, it can also cause the oxidative damage. So during the course of exposure to the heat, there are different oxidative radicals that are present or formed during this method of sterilization that ultimately kills the organism. And moreover, the heat is also said to be highly toxic in nature to the microorganism. They completely kill the organism due to its toxicity. So the mode of action in case of heat is due to protein denaturation. It can be oxidative damage or it can be toxic effect to the microorganism. So these are the three reasons why the organism usually dies when they are exposed to the heat. Now this heat can be applied either in a dry form or a moist heat form. So again, on the conditions, depending on the material that is to be sterilized, you can go for dry heat sterilization or moist heat sterilization. So in comparison to dry heat and moist heat is more effective because it has got more penetration power when compared to that of the dry heat, which is a bad conductor of heat. So moist heat is more effective when compared to that of the dry heat, and the units that are measured when you are using heat as a mode of sterilization is called as thermal death point units. That is the amount of time required to kill a suspension of microorganism at a predetermined temperature in a specified environment. That is, you are killing a suspension of cell in a particular time at already set temperature. So this thermal death point will vary from one organism to the other organism depending upon what type of culture you are using, whether it is an young culture or whether it is a old culture, whether it is a dormant one or whether it is an actively growing one, whether it is a vegetative form or a sporulating form, because each organism has its own way of surviving in nature, vegetative or actively growing. So you can easily kill the organism when they are exposed to the heat, whereas sporulating forms, they are resistant form, they are not easily killed by the heat. So moist heat is the best of choice to eliminate the microorganism when you are using organisms such as sporulating organism. So heat can be applied in the form of two methods, dry heat sterilization and moist heat sterilization. So before selecting heat as a mode of sterilization, we should remember certain points so that we can increase the effectiveness of the heat to kill the microorganism. So the factor that influences the sterilization by heat, that is, that renders it either inactive or it increases the efficiency of the heat, are nothing but the type of heat that we are using, whether it is a dry heat or a moist heat. As already discussed, moist heat is more rapid and has good penetration power. Next is the time and temperature, already discussed, that is they are directly inversely proportional, so it can be less time more temperature or more time less temperature, and again depending on the organism, whether it is a vegetative form or a sporulating form. Next we have the number of organisms, that is called as biological burden present in a particular environment. If the number of organisms are less, then it takes less time to kill the organism and less heat to kill the organism. Whereas if the biological burden is more, then it takes more amount of time and more amount of temperature to kill the organism. Now coming to the characteristics of organism, as already discussed, whether they are vegetative forms or a sporulating form, whether they are actively growing or in a dormant in nature, whether they have capsules, whether they have an external appendages that prevents them from the heat. So you have to know which type of organism you are going to eliminate from a particular substance. And finally the environment in which you are going to carry out this sterilization process. So you should keep in mind that there are no organic debris that are present in that particular environment, because this organic debris usually protects the organism from any type of heat that you are going to expose to kill the organism. So they will not allow the heat to penetrate to the organism or reach the organism, thereby making it inactive in nature or ineffective in nature. So these are the factors that are to be kept in mind or bore in mind before selecting any type of sterilization, most probably the heat, whether it is a moist heat or a dry heat. Now coming to the dry heat of sterilization. So dry heat is the one that itself indicates that there is no moisture present in it, that is no water or moisture present in it. So it is completely a dry heat, and the methods that are used in dry heat are flaming, incineration. We have a specialized chamber or an oven which is called as hot air oven, that is mostly used to sterilize labware or the glassware. So coming to the flaming is the method wherein we sterilize the inoculation loop and the spatulas in the microbiological laboratory. The inoculation loops are the ones that are used to transfer culture in microbiology. That is, with the help of the inoculation loop, you can take up the culture. And this loop is specially made up of nichrome wire that gets easily sterilized when exposed to the Bunsen burner. So remember, when we are exposing the inoculation loop to the Bunsen burner, it is at the blue flame that we are going to eliminate the microorganism. So apart from that, if there are any organism already present on the loop, then you can disinfect it by dipping it in the disinfectant such as isopropyl alcohol and then expose it to the flame, so that they are completely eliminating the organism. And with the help of this, you are preventing the dispersion of microorganism in the environment when they are directly exposed to the heat. So that is the method of choice to eliminate the organism by directly exposing to the Bunsen burner, which is called as flaming. The next method is called as incineration. Incineration is directly burning the material with the organism. So mostly used to eliminate organism from the contaminated clothes, animal carcass and pathogenic material, and the material that are mostly used to carry out experiments in the laboratory or the experimental labs. They are usually eliminated or killed with the help of incineration. Take for example the laboratory animals, guinea pigs and all, when they die, they go about with the incineration technique to eliminate the organism as well as the bodies of the particular experimental animal. So these are the two methods, and the third one is called as hot air oven. It is a specially designed oven that is used to bring about sterilization of different labwares in the microbiology laboratory, and mostly used to sterilize glasswares, forceps, scissors, scalpels, syringes, swabs, pharmaceutical products. And again, you have to remember in mind which type of product you are going to sterilize, and the temperature in the hot air oven is around 160 degrees centigrade for 1 to 2 hours, depending upon the nature of the material that is getting sterilized. And this oven is made up of it has an electrical heating element that transfers the heat, and there is a fan circulating inside that circulates the air inside the hot air oven, and this fan distributes the equal amount of air inside the hot air oven. So when we are preparing to make any sterilization in the hot air oven, we have to see that glasswares are completely dry, because presence of water might crack that particular glassware. So keep the glassware in a dry condition and see to it that the hot air oven is not overloaded, because the circulation of air will not reach to each and every part of the particular object. So it should not be overloaded, and it should be equally distributed, so that the dry heat is being circulated equally inside the hot air oven and killing the microorganism. And moreover, this rubbers and the pharmaceutical products or antibiotics cannot be sterilized with the help of dry heat, because they are heat sensitive in nature, and dry heat is a bad conductor of heat, so it has less penetration power. So you can move ahead with the other type of sterilization, that is called as moist heat sterilization. So here in the moist heat, as already discussed, dry heat and moist heat are the two ways that can be used to sterilize particular object, and moist heat is more effective when compared to that of the dry heat because of its penetration power. Moist heat as itself says that there is a steam or there is a moisture that is present. So when this moist heat or the moisture that is present usually goes and falls on a particular substance, and they increase the temperature of that and thereby kills the microorganism. So moist heat is more preferred choice than the dry heat, but there are certain components that cannot be sterilized with the help of moist heat, and that can be sterilized only with the help of the dry heat. Now coming to the moist heat can be applied in the form of three types. One is temperature below 100 degrees centigrade, temperature at 100 degrees centigrade, and temperature above 100 degrees centigrade. So the temperature below 100 degrees centigrade, the method of choice is pasteurization. As we all know, pasteurization is mostly used to eliminate organism from a particular product or the substance that is called as a milk. So here pasteurization is eliminating the vegetative forms of the organism. The temperature applied in pasteurization is around 63 degrees centigrade to 30 minutes, or it can be flash pasteurization, that is around 72 degrees to 15 seconds. So this is called as flash pasteurization. So you can see the temperature over here. Temperature is very high and the time is very low, and then immediately it is cooled to around 13 degrees centigrade. So here in this case we are not increasing the temperature above 100 degrees because they may cause the protein coagulation. So that are most important part of the milk. So we have to see to it that we are not destroying any quality of the milk. We are increasing the temperature to eliminate most of the organism, but we are not destroying the quality of the milk. That is in pasteurization. So eliminates most of the organism except Coxiella burnetii, that is mostly seen in the milk and causes different types of infections in the human body. But mostly with the help of pasteurization effect, you can kill most of the organisms. And next coming to temperature at 100 degrees centigrade, the phenomena that are used are nothing but boiling and tyndallization. So boiling is making the substance or the water to boil at 100 degrees centigrade, and you can sterilize or eliminate most of the organisms which are vegetative in nature, but you cannot eliminate the sporulating form. And boiling is not a choice for sterilizing surgical instruments, so you cannot go for sterilizing the surgical instrument in boiling, but you can eliminate only the vegetative forms. Hot air oven is another important instrument used in dry heat sterilization. So it is a chamber where we can use to sterilize different types of lab waste such as glassware, forceps, scissors, scalpel, syringes, swabs, and some of the pharmaceutical agents. So here in the hot air oven, it is a chamber that is usually run by electricity and that provides the heat inside the chamber, and the heat that is provided is called as a dry heat, and the dry heat is exposed to different material that kills the microorganism. So when preparing the materials materials to keep in the dry heat sterilization, we have to see that the glass test tubes or any beakers are completely free from moisture. Otherwise, they will crack the material. So, before that, you have to make it dry and then keep it in the hot air oven. You cannot sterilize rubber in the hot air oven, and certain materials such as antibiotic vaccines can also be not sterilized in hot air oven, because the temperature in hot air oven is around 160 degrees centigrade for 1 hour, and mostly is a preferred choice to sterilize most of the lab waste. And the best method to identify whether the sterilization is accomplished or not in a hot air oven is making use of Clostridium spores that are impregnated on strips and then placed it in the hot air oven. And after sterilization is completed, that is 160 degrees for 1 hour, you can remove that and inoculate onto the thioglycolate or a cooked meat media and then incubate it for 5 days and see for the growth of organism. So if the growth is there, that means the sterilization is not accomplished. As already discussed, a substance can either be completely sterilized or it is not sterilized. So in that case, you can identify whether hot air oven can be used to sterilize different material or not. And there are certain materials that cannot be sterilized with the help of dry heat. So move on with the moist heat of sterilization. So here the moist heat, as already discussed, it contains moisture, and when this moisture or the moist heat when falls on a particular substance, it gives away its latent heat and it raises the temperature of that particular substance and kills the microorganism. Most preferred choice is the moist heat than the dry heat, because it has more penetrating power and it is rapid in nature in its killing. So moist heat can be applied in 3 forms. One is temperature below 100 degrees centigrade, temperature at 100 degrees centigrade, and temperature above 100 degrees centigrade. So in case of temperature below 100 degrees centigrade, the phenomena that is used is called as pasteurization. Pasteurization is heating of a particular substance such as milk to an extent that you are eliminate the microorganism, but you are not destroying the quality of the milk. So here you are going to eliminate most of the organism at 60 degrees centigrade for 20 minutes, or it is 72 degrees centigrade for only 15 seconds and immediately cooling it to around 13 degrees centigrade. So in this particular procedure, we are killing most of the organisms such as Brucella, Mycobacteria, etc., except a particular organism which is called as Coxiella burnetii. So this is the organism that causes the spoilage of the milk. So apart from that, you can kill most of the organism in case of sterilization, that is the pasteurization. The next method of choice is at 100 degrees centigrade, mostly involving boiling or tendalization. Boiling is the method that is used to boil water and kill most of the vegetative organism from particular substance. You cannot eliminate organism from surgical instrument, and it is not a choice for sterilizing surgical instrument in boiling, but only to see that they are eliminating the vegetative form, because sporulating forms are most heat resistant. You have to increase the temperature to kill the organism. So in case of boiling, you are only eliminating the vegetative forms. So next part is called as tendalization. Tendalization is usually nothing but steam sterilization. It was proposed by Koch and Arnold, and a steamer is developed by them, where we can sterilize most of the media in this particular steam sterilization. So the temperature that is taken up in tendalization or in Arnold or Koch sterilizer is 90 degrees centigrade. But if the media is of sugar or glycogen, you have to increase the temperature to around 100 degrees centigrade with particular time to kill a particular type of microorganism. So tendalization is a process that is carried out for 3 successive days. So the logic behind this is, when you are exposing the organism to 100 degrees centigrade for 30 minutes on a first day, you are killing most of the vegetative form. So next successive day, again you have to keep for incubation. On the successive day, that is the next day, this sporulating forms that are present will germinate into the vegetative form. So on the second day, you are killing the sporulating organism that has developed into vegetative cells, and on the third day, again you are completely eliminating the organism. So that is called as partial sterilization or fractional sterilization, also called as sterilization. So 3 consecutive days eliminating the microorganism along with the sporulating forms. And the next the moist heat sterilization is called as temperature above 100 degrees centigrade, and the instrument that is used is autoclave. So the best method of choice again in the microbiological laboratory to sterilize the media and other components. So autoclave usually works on a principle of steam sterilization or steam under pressure. So as the pressure increases, the temperature also increases. As we all know that bacterial spores are highly resistant in nature. You cannot kill them either with a dry heat or a moist heat that is at 100 degrees centigrade. So you have to increase the temperature to around 120 degrees centigrade to kill the organism, and that particular 120 degrees centigrade is accomplished in a autoclave. So it is a cylindrical or a horizontal cylinder consisting of different foils that provides the electricity, and it develops a steam. You add a water into this chamber to develop the steam, and the steam that is usually produced in the autoclave is regulated by the pressure. So we are increasing the pressure by closing the lid and increasing the pressure to around 15 lbs. At 15 lbs, the temperature of the steam will be around 120 degrees centigrade, and when this steam falls on the microorganism, it gives away its latent heat, increasing the temperature of that particular organism and thereby killing the organism. That is what is called as steam under pressure. Steam is under pressure of 15 lbs and the temperature is around 120 degrees centigrade. So you have to hold this particular 120 degrees centigrade at a time of around 15 to 20 minutes. That is called as a holder time. So at this particular time you are eliminating most of the organism. So again when you are using the autoclave as a source of sterilization, you have to see that everything is packed properly and then kept, and the lid is tightly closed. The lid consists different gauges. One is to tighten up the lid so that no steam comes out from the autoclave. You have a pressure gauge to know the amount of pressure that is present, and you have a steam valve to release the pressure whenever required. So before opening up the lid, you have to be very careful. Once the autoclave gets cool, leave it for some time and then you have to open it. Otherwise the steam directly will cause a harm to the person who is usually operating it. So whatever the material that has to be sterilized has to be completely packed and then placed it in the autoclave. So mostly autoclave is used to sterilize the microbiological media that is used to grow microorganisms in the lab. And the sterilization control, as we have seen in the dry heat sterilization where Clostridium was used as an indicator organism, here we have Bacillus stereothermophilus, which is a most heat resistant microorganism producing spores. So if this organism is killed, that means all other organisms can be killed. So here also we can do the same thing. Impregnate strips with the help of Bacillus stereothermophilus, keep it in an envelope and then place it in between certain components in the autoclave and go for sterilization. And after sterilization is complete, go for incubation of this particular organism on a particular media, that is the specific media for the organism, and then see for the growth. So in that case you can know whether the sterilization is accomplished or not. So it is the dry heat or the moist heat that can be used in the laboratory to eliminate the microorganism. The next most reliable method to eliminate microorganism but not kill microorganism is filtration. So here we remove the bacteria from heat labile materials. Heat labile materials are the ones that are very sensitive to heat. You cannot expose them to either dry heat or moist heat to eliminate the organism. But there will be certain organisms that are present on this that may cause the contamination of that particular product. So you have to remove the organism. So filtration is mostly used to remove organism from the heat labile fluids. So there are different filters that are used in a microbiological laboratory to eliminate the microorganism. And filtration is a choice to remove microorganism from antibiotic, vaccines and certain pharmaceutical products, drugs, etc. that are highly heat labile in nature. And the filters that are used in microbiological laboratories are called as candle filters, asbestos filters, sintered filters and membrane filters. And the mostly used to purify filters are the candle filters. So mostly used for the purification of water, and they can be unglazed filters, example Chamberlain. They can be diatomaceous earth filters that are called as Bergfield filters, and then we have asbestos filters, which are the one use single use type of filters. You can use them and then you can throw away, but because of certain carcinogenic materials that are present in asbestos, you are hardly using this type of filters. Examples of asbestos filters are Seeds filter and Sterimat, but they have good adsorbing capacity. Adsorbing capacity is to retain the bacteria when a particular solution is being filtered. And then you have sintered glass filters that are usually fused at very high temperature of glass particles of graded size. And then last one is called as a membrane filters. So the other type of filters that are used in microbiological laboratory are the membrane filters, which are made up of cellulose esters and polymers, and these are the one that retains most of the bacteria and sometimes even the viruses. So membrane filters are mostly used in microbiological laboratory to test the sterility and also to purify the waters. And this membrane filters also helps us to know the type of organism when they are incubated along with the media. The next method of choice physical method of sterilization is the radiations. Radiations are high energy waves, and they usually causes either ionizing effect or non-ionizing effect in a particular object on which they are usually striking, mostly the microorganism. So here the radiation can be non-ionizing. That means they are not forming any ions such as electrons or hydroxyl ions or nascent oxygen. Okay, so this non-ionizing radiations, examples are infrared radiations and UV radiations. Mostly UV radiations, as already discussed in sunlight, they have a deleterious effect on the microorganism, and they combine with the DNA of the organism and they form the thiamine dimer. So mostly used in enclosed areas to sterilize a particular area with the help of UV chambers. We have this laminar airflow chamber where we carry out the pure culture techniques in microbiological laboratory. So inside the laminar airflow chamber we have a UV light. So before carrying out any technique, what we do is we on the UV light for a few seconds and then we switch it off and then carry on with the other procedures. And during that particular period of time, the UV light usually eliminates most of the organism present in that particular enclosed areas. Now next methods radiations are ionizing radiations. Example X-rays, we have gamma rays, we have cosmic rays, etc. So the mode of action of this ionizing radiation is that they forms the ions, or they knock away the electron and they form the ions such as hydroxyl ions and nascent oxygen. Now when these ions goes and interacts with certain components of the bacteria, they kill the bacteria. So X-rays, gamma rays and cosmic rays are mostly used as sterilizing agents, and these radiations, when they are used to sterilize foods, they are called as cold sterilization, or they are called as radapodization. Okay, and next thing, the last thing in a physical method of sterilization is ultrasonic or sonic vibrations. So ultrasonic waves are nothing but about 15,000 cycles per second. They causes bacteria to cavitate. That means they rupture the cell wall of the organism, thereby kills the organism. But this is not the best method of choice in microbiology to kill the organism. Mostly used by the dentist to clean the teeth. The instrument that is called as a cavitron, and here it is called as a cold boiling because it is not increasing the temperature of that particular liquid in which it is usually carried out. So that is called as cold boiling. So these are the different methods of choices when we adopt physical methods of sterilization. They are sunlight, we have that is the drying, we have heat that can be applied either in the form of a dry heat or a moist heat, and then we have filtration that removes the microorganism but do not kill the microorganism, and we have radiations, and ultimately it is the ultrasonic and the sonic vibration. So next, coming to the chemical methods of sterilization. So as already discussed, sterilization and disinfections are the same in their purpose, but on which material we are carrying out usually varies. So here we have got our wide variety of chemical agents that are having both bactericidal as well as bacteriostatic effect. So the concentration of chemical that we are using will show whether it is bactericidal or bacteriostatic in nature. So amount or the concentration, when it is more, it can lead to the killing of organism, and the amount or the concentration is less, it brings about a bacteriostatic nature in the organism. Now when you are choosing a chemical for a chemical method of sterilization, you should know which is the ideal chemical agent. So ideal chemical agent usually satisfies certain properties, only after which you can use it to bring about the disinfection. So ideal antiseptic or disinfectant should have certain properties. The first important property is it should be a broad spectrum and narrow spectrum. Narrow spectrum is the one that kills only few types of microorganisms, but broad spectrum antibiotics or broad spectrum chemical agents brings about killing of most of the organism, including the spores. So when you are selecting a chemical, you have to see that it is a broad spectrum chemical agent. Next is it should be active in the presence of organic material. So already discussed, the organic material might protect the organisms, thereby rendering it inactive. So when you are selecting a chemical agent, it should see to it that it is active in nature, even it is present in the presence of a organic material. Next is it is effective in all conditions, irrespective of what the condition is present in the environment, whether it is a temperature or a pH variation. The chemical agent should be active in or effective in all condition. Next is it should be rapid in action. It should not take long time to act on the organism, because most of these chemical agents are used to bring about sterilization of certain areas. So you have to see to it that it is rapid in its action, and it should kill the organism as soon as possible, and it should have high penetrating power, as is we have seen in the moist heat. Because it has a good penetrating power, it can penetrate the organism and kill the organism. Similarly, the chemical agent should penetrate into the organism and bring about the killing of organism, and it should not be very expensive. It should be cheap in nature, so that everybody can afford it and bring about the sterilization process. And it should not have any toxic effect on the humans, no deleterious effect. It should be active only towards the organism, but it should not be active towards any other component that is in vicinity of it. And finally, it should be stable enough, because the time of entry of the chemical agent in that particular medium to reach the organism, it will take some time. So during that time, the antibiotic or the chemical usually becomes ineffective in nature. So it should not lose its effectivity till it reaches the organism and kills the organism. So that is what is called as stability. The material should be stable enough to kill the microorganism, even in the presence of any other material. So this is the ideal chemical agent or the chemical antiseptic that is mostly used when they are used in bringing about the disinfection process. Now coming to the mode of action, most of these chemical agent kills the microorganism either by protein coagulation. So we can have two procedures over here. One is protein denaturation, where the tertiary structure of the protein is completely denaturated, whereas here protein coagulation, they precipitate the protein, they make the protein inactive to take part in the biological activity. So that is what is called as protein coagulation. Next is they disrupt the cell membrane. As we all know, cell membrane is the most important component of the organism, which has selective property, also called as semi-permeable membrane. So once that selective permeability is lost, the organism is rendered dead or killed either due to plasmolysis or osmolysis. Next is removal of free sulfhydryl groups from enzymes. Now enzymes are the most important biocatalysts that helps to carry out biochemical reactions inside the organism. So wherever there is a free sulfhydryl group, these chemical agents, they usually remove the sulfhydryl group, making the enzyme inactive in nature, thereby not making it participate in the biochemical reaction. And finally, they are some of the chemical agent can also act as substrate competitors. Substrate competitors is nothing but enzyme when reacts with a particular substrate, they give rise to products. So instead of that particular substrate, the chemical goes and binds. As a result of which, the product is not formed in the organism, that is very important for its biological activity. So these are the different modes of actions of how the chemical agent usually kills the microorganisms. The mode of actions of this chemical agent is protein coagulation, disruption of cell membrane, removal of free sulfhydryl group from enzyme, and finally they act as some substrate competitors. So protein coagulation is they precipitate the protein inside the microorganism so that no longer these proteins are involved in biochemical activities. Apart from that, they disrupt the cell membrane. Cell membrane has a selective toxicity. That means they will allow only certain components to move in and out of the bacteria. So when this semi-permeability is lost, the organism dies either due to plasmolysis or osmolysis. Apart from that, this chemical agent can also target free sulfhydryl groups of an enzyme. Enzyme is an important biocatalyst to carry out different biochemical reaction inside the microorganism. So when this sulfhydryl group is removed or it is being targeted by the chemical agent, the enzyme is rendered inactive and it is no longer taking part in the biochemical activities. And finally, these chemical agents can also act as substrate competitors. So when enzyme, in a normal condition, when an enzyme binds to a particular substrate, they form a product. So here in the case of this substrate competition, the chemical agent usually goes and binds to the enzyme in place of substrate, and the product that should be formed is not formed. Ultimately, the organism gets killed. So these are the different mode of action of the chemical agent. And the most important types of chemical agents that are used in chemical methods of sterilizations or as disinfectants are alcohol. The most prominent one is the alcohol. Examples of the alcohols, mostly the lower alcohols are used as disinfectants rather than the higher alcohols, which are very toxic in nature. So the lower alcohols mostly that are used are methyl alcohol, ethyl alcohol, we have isopropyl alcohol, and these alcohols are mostly used as skin antiseptics. And the isopropyl alcohol is mostly effective against gram-negative organism because it is a fat solubilizer. Gram-negative organism on its cell wall has an extra component which is called as lipopolysaccharide layer that is easily dissolved by the alcohol. Now, because of that, the organisms die due to change in the porosity or due to the formation of holes on the cell wall. And then this particular alcohols is effective at 60 to 90% because 100% of alcohol cannot enter into the organism. So you have to dilute a little with the water, and it is only at 60 to 90%. Mostly it is 70% of the alcohol that is harmful to the microorganism. So here the disinfectants such as isopropyl alcohol is mostly used to treat the clinical thermometers or disinfect the clinical thermometers. So initially it was methyl alcohol that was used to as a disinfectant, but because of its toxic effect it is being avoided, but it is mostly active against the fungal spores. So next important chemical agents that are used in chemical method of sterilization are the aldehydes. Aldehydes are two types. One is a formaldehyde, the other one is a glutaraldehyde. Formaldehyde usually is present in two forms. Either you can use it in the gaseous form or in an aqueous form. So it is mostly active against the amino group of protein molecules. So protein molecule is made up of amino group and the carboxyl group. So here the formaldehyde usually targets the amino group. It removes the amino groups and makes the protein inactive in nature or denatures the protein. So aqueous solution of this formaldehyde is usually available in the commercial departments in the name of formalin. So here it is both bactericidal as well as sporicidal in nature. So effective against the bacterial spores also. And formaldehyde can also be used to preserve anatomical specimens and used to sterilize the surgical instruments such as heat sensitive catheters, fumigate. They are used to fumigate wards and labs also. So this is more toxic when compared to the glutaraldehyde. The vapors of the formaldehyde is irritant and toxic. So you can nullify the irritation of this formaldehyde by smelling with the ammonia. So next important type of aldehyde is a glutaraldehyde. Glutaraldehyde is a higher form of the aldehyde groups, and it is effective on tubercle bacteria also as well as fungi and viruses. So more having effective than the formaldehyde, and it is mostly used to sterilize cystoscopes, bronchoscopes, face masks, endotracheal instruments, polythene tubings, etc. So less irritant when compared to that of the formaldehyde and less toxic also. The next type of chemical agents that are used are dyes. So dyes are of two types. They are acridine dyes and aniline dyes. Mostly these dyes are used in microbiology to stain the microorganism, but they do have certain bactericidal activity. They bring about the killing of microorganism. So we have acridine dyes and aniline dyes. Example of acridine dyes are brilliant green, malachite green, crystal violet, etc. And these dyes are mostly active against the gram-positive organism which have acid groups in their cell wall. They target the acid group on the cell wall, thereby forming pores on the cell wall and killing the microorganism. And these acridine dyes are mostly used in a selective media to select or allow the growth of only selective group of organisms. So here in the selective media, what we do is, selective media means selecting particular group of organism or allowing the growth of only that particular group of organism, whereas inhibiting the other organism. So when we add certain type of dyes, they inhibit the other organism, whereas the nutrition that is provided will allow only particular group of microorganisms. So that is what is called as selective media. So these dyes are mostly used in the selective media to inhibit the group of microorganisms. Now coming to the aniline dyes, example is proflavin, acreflavin, euflavin, aminacrin. So these organisms are active against the gram-negative bacteria. They impair the DNA of the organism, or the DNA is the most important component that helps the bacteria to reproduce. So once it gets impaired, then there is no longer activity in the bacteria and the organism, they will die. And the other group is halogens. Halogens are also mostly used to bring about sterilization, and some of the halogens are used as antiseptic, whereas some are used to clean the swimming pools or the purification of water. So here we have two important halogens that are mostly used. One is called as the iodine, the other one is the chlorine. So iodine mostly initially it has been used since centuries as a skin disinfectant, but because of the staining property of iodine, that has been a bit modified by combining with the organic molecule which are called as iodophores. So these iodophores are highly bactericidal in nature and can be applied to eliminate organism from the skin. Now coming to the chlorine, chlorine is mostly used to disinfect the water supplies. They are used to disinfect swimming pools and food and dairy industries. So the form of chlorine that are used are hypochlorites and chloramides that are highly active against the microorganisms. So this hypochlorite and chloramides usually form a nascent oxygen, and this nascent oxygen goes and interacts with certain component of the organism and kills the organism. The next important components that have been used in centuries are phenols. So this phenol is usually obtained by distillation of the coal tar at a temperature of 170 to 190 degrees centigrade. And phenol's mode of action is to damage the cell wall or the cell membrane of the organism, or they usually precipitate the proteins, and they can also inactivate the membrane bound oxidases and dehydrogenases. So these membrane bound oxidases and dehydrogenases are the one that helps to produce ATP during the oxidative phosphorylation. But because of the toxic effect of the phenols, they are no longer able to get activated, and thereby the organism gets killed. So phenols have been the best choice by the Joseph Lister. He used it to prevent sepsis during his surgery. So since then, phenols and various compounds of the phenols are being used to disinfect a particular object. So we have different types of phenols that are commercially available right now. It's called as Lysol, Cresol, hexachlorophene, chlorhexidine. So mostly used to eliminate from a particular area. Now the next one are the gases. So gases can be used as a disinfectant to disinfect a particular enclosed areas such as bookshelves or laboratories or clothing, etc. So here we have got three different types of gases that are used as chemical disinfectant. One is an ethylene oxide, you have beta propionylactone, and then finally formaldehyde. That, as we have already studied, that formaldehyde can be used either in the form of a liquid or in the form of a gas. So in the form of a liquid it is formalin. In the form of a gas, this is mostly used to sterilize the enclosed area of the laboratory. Now coming to ethylene oxide, the most important one in the gases highly toxic and inflammable in nature. So the colorless liquid has a boiling point of around 10.7 degrees centigrade, above which it becomes completely volatile in nature. But the mode of action of this particular ethylene oxide is it alkalides amino hydroxyl, and then you have carboxyl group that is present in the enzyme and inhibits the enzyme activity. Apart from that, it can also act with the DNA and the RNA, causing changes in the DNA and RNA and no longer able to reproduce. And finally, this can be used to sterilize heart-lung machines. We have respirators, dental equipments, books, clothing, etc. So there is steam sterilizer, that is a gas sterilizer, which is called as ethylene oxide sterilizer, in which all the equipments of the surgicals are kept and they are sterilized in the hospitals. And then the other important gas that is used is called as beta propione lactone. Here the boiling point is around 163 degrees centigrade, and 0.2% of BPL is mostly used as a sterilizing agent, but it is more toxic and more volatile in nature, so it is mostly avoided. And then formaldehyde. The next chemical disinfectants are the surface active agent. As the name itself indicates, they act on the surface of microorganism, mostly targeting the phosphate group of the microorganism, losing the cell wall or the cell wall permeability. So here these surface active agents are mostly the detergents that we use in day-to-day life to eliminate the microorganism. And they can be classified into 4 types. One is the anionic, cationic, non-ionic and amphoteric in nature. So among the 4, it is the cationic that is most preferred choice to eliminate the microorganism. So act on the phosphate group, and they also loses the semi-permeability of the organism. And the example of this are quaternary ammonium compounds. Example, acetyl methyl ammonium bromide, which is available as cetabolone or cetrimide, and benzyl choline chloride. And apart from this, anionic examples are detergents, and we have amphoteric compounds which are called as TEGO compounds. So all of these usually targets the cell wall of the organism and kills the organism. And the last one is the metallic salts. So high metal can be used to eliminate the microorganism and have been used since many times or longer period of time as a germicidal agent. So salts of heavy metals such as silver, copper and mercury are mostly used. And the mode of action of this metallic salt is, again, they target the protein, they coagulate the protein, they combine with the sulfhydryl groups of the enzyme, making it inactive. And the organic compounds such as thiomersal, phenyl mercuric nitrate, mercurochrome are usually bacteriostatic in nature. That is, they do not kill the organism but inhibit the activity of the microorganism. And finally, the copper salts are mostly used as fungicides in nature. That is, they have the ability to kill the microorganisms. So whatever we have these creams, if a person is suffering from any fungal infections, you can see a minute concentration of the copper salt that inhibits the growth of the fungus. So this is all about the physical and the chemical method of sterilization, the objective of which is to completely eliminate the microorganism from a substrate.